In the previous session, we understood what is mutation and their classification. In this session, we will understand what are mutagens. So, mutagens are the substances or agents which induce artificial mutation. They are of basically three types: physical mutagens, chemical mutagens, and biological mutagens. Under physical mutagens, we have radiation and heat or temperature. Under chemical mutagens, we have base analog, intercalating agent, alkylating agent, deaminating agent, and metals. Under biological mutagens, we have transposons, virus, and bacteria. Physical mutagens, it is radiation. The radiations are of two types. One, the ionizing radiations like the X-rays and gamma rays, and the other, the non-ionizing radiations like ultraviolet rays and visible light. Ionizing radiations. The charges are usually neutral, electrically neutral. When these ionizing radiations pass through matter, they dissipate their energy into the electrons from the outer shell of atom, and there is a loss in their balance, and negatively charged electron leaves atom and is no longer neutral but becomes positive. This positively charged atom is called ion. The ejected electron move at high speed, knock other electron free from their respective atom, and attach to other atoms and convert the ion into negatively charged ions to achieve their stable configuration. That is, neutral charge ion undergo many chemical reactions, and during these chemical reactions, ionizing radiation is thought to cause mutation. These ionizing radiation cause break in polysugar phosphate backbone of DNA, causing chromosomal mutations such as breaks, deletions, additions, inversions, and translocation. Non-ionizing radiation. The ultraviolet light is a non-ionizing radiation which may cause mutation. The wavelength of around 2600 Armstrong is the most effective wavelength for mutation. The substance absorbs sufficient energy from UV light. Some of their electrons are raised to higher energy levels and is called excitation state. The excited molecule becomes reactive and mutated and is called photoproducts. The ultraviolet radiation produces several effects on DNA, one being the formation of chemical bonds between two adjacent pyrimidine molecules. The two thymine residues associate or dimerize to form a dimer. The position in the DNA helix becomes so displaced that they can no longer form hydrogen bond with opposing purine. This effect is called dimerization and interferes with the proper base pairing of thymine with adenine and may result in thymine pairing with guanine. Temperature as mutagen. The rate of all chemical reactions are influenced by temperature. It is not surprisingly that temperature can be mutagenic. For example, an increase in 10 degrees Celsius temperature increases the mutation rate 2 or 3 folds. Temperature probably affects both thermal stability of DNA and the rate of reaction of other substances with DNA. Chemical mutagen. Many chemical substances have been responsible to increase the mutation in genes. The ability of chemicals to induce mutation was first demonstrated by Auerbach and Robson in 1947 on the experiments with male Drosophila melanogaster. Any chemical substance that affects the chemical environment of a chromosome is likely to influence, at least indirectly, the stability of DNA and its ability to replicate without error. It can affect chromosomal DNA in following two ways: direct gene change and copy error. Direct gene change. Chemical mutagens affect DNA directly. They affect the constituent of DNA only when DNA is not replicating. Example, nitrous acid converts adenine into hypoxanthine and cytosine to uracil by deamination. Like the nitrous acid, nitrogen mustard, formaldehyde, epoxy sites, dimethyl and diethyl sulfonates have also direct mutagenic effect on DNA molecule. Copy error. Certain chemical compounds called base analogs closely resemble with certain DNA bases and are therefore act like mutagens. During DNA replication, they are incorporated by DNA in the place of the normal DNA bases, and these are referred to as base analogs. These base analogs are substances having structural similar molecular structure as that of DNA base and are available. These analogs may be incorporated into the replicating DNA strand. Example, 5-bromouracil. It is in its usual keto form is a structure analog for thymine and will substitute for thymine. The other example is aminopurine. It is undifferentiated purine that apparently can pair with cytosine and thymine. It is thought that 2-aminopurine acts by switching pyrimidine. Inhibition of precursors of nucleic acid. There are some mutagens which interfere with synthesis of nitrogen bases of nucleic acid, such as purines and pyrimidines. First one, deamination. Some chemical substances such as nitrous acid cause transitional mutation due to oxidative deamination. In this process, the amino group of the DNA base is replaced by hydroxyl OH group by the chemical mutagen. That is, the adenine is deaminated into an hypoxanthine by nitrous acid. By tautomeric shift, the hypoxanthine is converted into more common or ketotautomer, which pairs with cytosine. Deamination converts cytosine to uracil, which has pairing properties similar to thymine, and in such a case, G is to C pair would be changed into A-T pair. Hydroxylamine and hydrazine. When DNA is treated with hydroxylamine, its cytosine base is the strongest reacting base. It causes hydroxylation of cytosine at amino group, giving rise to hydroxyl cytosine. The hydrazine affects DNA by breaking of rings of uracil and cytosine, giving rise to pyrrozolone and 3-amino pyrrosol. The treatment of RNA or DNA with anhydrous hydrazine results in the destruction of their pyrimidines. Alkylating agent. Some alkylating agents carry 1, 2 or more alkyl group in a reactive form and act as strong mutagens. Example, dimethyl sulfite, dimethyl ethyl sulfate, methyl methane sulfonate. The mutagens produce mutation in the following ways. They add ethyl or methyl group to guanine. This makes guanine a base analog to adenine. To remove the alkylated guanine, this is known as depurination. The loss of the base produces gap in the DNA chain, which may be filled with a wrong base, thus producing mutation. The gap may also produce a deletion, causing mutation. Thank you for watching the video. If you like, please do share, like and subscribe to our channel. And for the detailed notes, you can click on the link in the description box below. Thank you.